Welcome to Veterinary Microbiology 151, Exercise number 4, Characterization of Bacterial Culture, presented by Group 3. For Part 1, we'll talking about characterization of bacterial culture in agar plates. First, we'll talk about pigmentation. Each distinct circular colony should represent an individual bacterial cell or group that has divided repeatedly. Being kept in one place, the resulting cells have accumulated to form a visible patch. Most bacterial colonies appear white, cream, or yellow in color and fairly circular in shape. Some examples are Bacillus subtilis, Proteus vulgaris, Staphylococcus aureus, and Streptococcus pyogenes. Second is the surface. Several terms that may be appropriate for describing the texture or consistency of bacterial growth. It's either dry or powdery, smooth or glistening, wrinkled or rugose, or mucoid. Some are contoured, with irregular or smoothly undulating, radiate, radius radiating from the center like spokes in a wheel, and concentric, which marked with rings having common center. Third is the shape of the colony, and it could be punctiform, circular, filamentous, rhizoid, spindle, or regular. Fourth is the size of colonies. It could be pinpoint, pinhead, or matchhead. Fifth is the margin of colonies. It could be entire, which have a smooth and doesn't have notches. It could be undulate, which have wavy or shallow indentations. Or it could be erase or serrate, which have irregularly notched. Or it could be filamentous, which have long, irregularly placed or interwoven threads. Or it could be curled, which have parallel chains in wavy strands. Or lobate, which have wavy or deeper indentations. Sixth, the elevation of colonies. This describes the side view of a colony, and these are the most common: flat, raised, convex, dome-shaped, ambinate, and umbilicate. Seventh is the odor. Microorganisms can produce different types of volatile compounds that may give characteristic smell, pleasant scent, or pungent odor. Production of these volatile chemicals depends on the metabolic characteristics of that particular organism. Sometimes scent or odor produced by bacteria can give important clues in the identification of microorganisms, but it is not a reliable rule. Some of the scents are unpleasant smell. It can be yeast-like smell, soil-like smell, rotten cooked fish odor, and sweet grape-like scent. It can also be ammoniacal smell, bleach-like odor, or caramel odor. 8 is the imparts pigment to surrounding medium. The type of agar can affect the imparts pigment to the surrounding medium, and here are some examples of the types of agar: triptych soy agar, chocolate agar, and McConkie agar.
Characterization of bacterial culture in agar slants. Agar slants can be used to culture bacterial cells for identification. Agar slants are useful in maintaining growth of microorganisms over long periods of time without allowing the cultures to dry out. Amount of growth. The amount of growth can be slanty or slight, moderate, abundant, or none. Pseudomonas aeruginosa is one of the examples of a free-living, gram-negative bacterium that exhibit rapid growth, facile genetics, and a large suite of virulence-related phenotypes. Color of colony. In a slant culture, the color of the colony is red because of the presence of lactose, glucose, and saccharose. Gram-positive bacteria like E. coli and Mebshella species are lactose fermenters and gives pink-colored colonies on McConkie agar medium. Consistency of agar slants can be butterous, viscid, membranous, or brittle. Agar slants are only in its liquid form. Optical characteristics. Optical characteristics of agar slants are opaque, translucent, opalescent, iridescent, fluorescent, dull, or glistening. These characteristics are all based on how much light travels through the colony. Form. Form of agar slants can be filiform, characterize growth along the line of inoculation. Spreading, which growth extends along the line inoculation. Equinolate or pointed margins. Beaded, which separates colonies. Rhizoid, a root-like structure, or arborescent, a tree-like growth. Odor. Is odor absent or resembling? In an ordinary medium like nutrient agar, P. aeruginosa produces a characteristic of grape-like odor of aminoacetophenol. Being a non-fermenter, the bacterium is also associated with acid production in culture rather than gas. Medium change. An example of medium change in agar slants are Simon citrate that is used to test an organism's ability to utilize citrate as a source of energy. Some type of bacteria that can grow on this medium to produce an enzyme called citrate permase that is capable of converting citrate to pyruvate and carbon dioxide. This test is done on slant medium because oxygen is necessary for positive utilization. So this carbon dioxide reacts with sodium and water to form sodium carbonate and alkaline product. This increases the pH above 7.6, which indicated bronchial blue that changes color from green to blue.
The characterization of bacterial culture in nutrient broth. Nutrient broth is typically made of a powdered beef extract that contains peptones, or broken down proteins. The powder is dissolved in water, put in test tubes, and sterilized. The examination of broth culture must include the following. First is the surface growth. Here we have flocculent, where small masses of growth of various shapes floating in the culture fluid. The ring. It has ring at upper margin adherent to glass. The pellicle. Growth in the form of a continuous or uninterrupted sheet over the surface. Lastly, the membranous. It is a thin coherent growth like a membrane. Second, the subsurface growth. We have the turbid, which is cloudy. Next, granular. It has small particles. The flocculent has small masses of various shapes. And the flaky has large particles. Here are the different growth patterns in broth. The clear or no growth, the turbid, meaning cloudy, the flocculent, pellicle, and sediment. Third, the amount of growth. None, no growth may be seen. Scanty means small or insufficient quantity. Moderate means average amount of growth. Abundant means large in quantity. Here's the sample image of bacterial cultures in broth media, wherein A is considered as none or no growth may be seen. B is scanty, C is moderate, and D is abundant. Next is the sediment, wherein granular means it has small granules. It is called flocculent if it has small masses of growth of various shapes. Flaky if it has separate flakes. And viscid if sediment rises as a coherent swirl. Next is the pigmentation. It is none or white. But some bacteria cause pigmentation into the broth, such as black and red, rather than white, which is the most common. The following are some examples of bacteria that was cultured broth. Bacillus subtilis, surface growth, flocculent, Subsurface growth flaky. Amount of growth moderate. Sediment flocculent. Pigmentation none. The Myobacterium smegmatis. Surface growth pellicle. Subsurface growth flocculent. The amount of growth moderate. The sediment granular. Pigmentation none. Lactococcus lactis. Surface growth flocculent. Subsurface growth flaky. The amount of growth moderate. Sediment granular. Pigmentation none. Staphylococcus epidermidis. Surface growth pellicle. Subsurface growth turbid. The amount of growth moderate. The sediment and pigmentation none.
Part 4. Characterization of bacterial culture in Aegir's land. Examination of broth culture must include the following. Number 1. Amount of growth. None. In Aegir's land, no growth can be visible. When there is no growth in the culture, there may have been a mistake throughout the process and you may need to repeat the experiment. Simple mistakes, like not letting the inoculating loop cool completely, can impact the culture, leaving you with no result in your Aegir's land. Scanty, amount inadequate or insufficient. Abundant, average amount of development to the point that the surface is hardly visible. Number 2. Form of growth. Filiform, uniform along the inoculation line. Undulate is a lumpy edge that bacteria can grow on. Small branch-like growth grows outward on the filiform edge. Wave-like layers stretch outward, form a curled border. The lobate edge is characterized by rounded finger-like growths that extend outward. Beaded, approximately halfway during inoculation, separate colonies. Papillate, entire papillate colonies of fish extract malt, extract agar. Moderate growth, brown, yellowish brown, smooth and whole, 1.2 mm in diameter. Villus, covered with villi. Arborescent, tree-like structure. Liquefaction, none or crateriform, bearing the appearance of a crater. Napiform, liquefaction in the shape of a turnip. Infundibulum, liquefaction in the shape of a funnel. Saccate, liquefaction in the form of sac. Stratiform, liquefaction layer creation in an organized manner.
And that ends our report. Thank you for watching.